Mechanisms of breathing vary among different groups of animals depending mainly on their habitats and levels of organization. Lower invertebrates like sponges, coelenterates, flatworms, etc. exchange O2 with CO2 by simple diffusion over their entire body surface. Earthworms use their moist cuticle, and insects have a network of tubes, tracheal tubes, to transport atmospheric air within the body. Special vascularized structures called gills, branchial respiration, are used by most of the aquatic arthropods and molluscs, whereas vascularized bags called lungs, pulmonary respiration, are used by the terrestrial forms for the exchange of gases. Among vertebrates, fishes use gills, whereas amphibians, reptiles, birds, and mammals respire through lungs. Amphibians like frogs can respire through their moist skin, cutaneous respiration, also. We have a pair of external nostrils opening out above the upper lips. It leads to a nasal chamber through the nasal passage. The nasal chamber opens into the pharynx, a portion of which is the common passage for food and air. The pharynx opens through the larynx region into the trachea. Larynx is a cartilaginous box which helps in sound production and hence called the sound box. During swallowing, glottis can be covered by a thin elastic cartilaginous flap called epiglottis to prevent the entry of food into the larynx. Trachea is a straight tube extending up to the mid-thoracic cavity, which divides at the level of fifth thoracic vertebra into a right and left primary bronchi. Each bronchi undergoes repeated divisions to form the secondary and tertiary bronchi and bronchioles, ending up in very thin terminal bronchioles. The tracheae, primary, secondary, and tertiary bronchi, and initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchiole gives rise to a number of very thin, irregular-walled and vascularized bag-like structures called alveoli. The branching network of bronchi, bronchioles, and alveoli comprise the lungs. We have two lungs which are covered by a double-layered pleura with pleural fluid between them. It reduces friction on the lung surface. The outer pleural membrane is in close contact with the thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The part starting with the external nostrils up to the terminal bronchioles constitute the conducting part, whereas the alveoli and their ducts form the respiratory or exchange part of the respiratory system. The conducting part transports the atmospheric air to the alveoli, clears it from foreign particles, humidifies, and also brings the air to body temperature. Exchange part is the site of actual diffusion of O2 and CO2 between blood and atmospheric air. The lungs are situated in the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by the vertebral column, ventrally by the sternum, laterally by the ribs, and on the lower side by the dome-shaped diaphragm. The anatomical setup of lungs in thorax is such that any change in the volume of the thoracic cavity will be reflected in the lung, pulmonary cavity. Such an arrangement is essential for breathing, as we cannot directly alter the pulmonary volume. Respiration involves the following steps: breathing or pulmonary ventilation, by which atmospheric air is drawn in and CO2-rich alveolar air is released out. Diffusion of gases, O2 and CO2, across alveolar membrane. Transport of gases by the blood. Diffusion of O2 and CO2 between blood and tissues. Utilization of O2 by the cells for catabolic reactions and resultant release of CO2. Breathing involves two stages: inspiration, during which atmospheric air is drawn in, and expiration, by which the alveolar air is released out. The movement of air into and out of the lungs is carried out by creating a pressure gradient between the lungs and the atmosphere. Inspiration can occur if the pressure within the lungs, intrapulmonary pressure, is less than the atmospheric pressure, i.e., there is a negative pressure in the lungs with respect to atmospheric pressure. Similarly, expiration takes place when the intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and a specialized set of muscles, external and internal intercostals between the ribs, help in generation of such gradients. Inspiration is initiated by the contraction of diaphragm, which increases the volume of thoracic chamber in the antero-posterior axis. The contraction of external intercostal muscles lifts up the ribs and the sternum, causing an increase in the volume of the thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs, i.e., inspiration. Relaxation of the diaphragm and the intercostal muscles returns the diaphragm and sternum to their normal positions and reduces the thoracic volume and thereby the pulmonary volume. This leads to an increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs, i.e., expiration. We have the ability to increase the strength of inspiration and expiration with the help of additional muscles in the abdomen. On an average, a healthy human breathes 12 to 16 times a minute. The volume of air involved in breathing movements can be estimated by using a spirometer, which helps in clinical assessment of pulmonary functions. Tidal volume, TV. Volume of air inspired or expired during a normal respiration. It is approx. 500 mmol, i.e., a healthy man can inspire or expire approximately 6,000 to 8,000 milliliters of air per minute. Inspiratory reserve volume, IRV. Additional volume of air, a person can inspire by a forcible inspiration. This averages 2,500 ml to 3,000 mmol. Expiratory reserve volume, ERV. Additional volume of air, a person can expire by a forcible expiration. This averages 1,000. Residual volume, RV. Volume of air remaining in the lungs even after a forcible expiration. This averages 1,100 milliliter to 1,200 milliliter. By adding up a few respiratory volumes described above, one can derive various pulmonary capacities, which can be used in clinical diagnosis. Inspiratory capacity, IC. Total volume of air a person can inspire after a normal expiration. This includes tidal volume and inspiratory reserve volume, TV plus IRV. Expiratory capacity, EC. Total volume of air a person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume, TV plus RV. Functional residual capacity, FRC. Volume of air that will remain in the lungs after a normal expiration. This includes ERV plus RV. Vital capacity, VC. The maximum volume of air a person can breathe in after a forced expiration. This includes ERV, TV, and IRV, or the maximum volume of air a person can breathe out after a forced inspiration. Total lung capacity, TLC. Total volume of air accommodated in the lungs at the end of a forced inspiration. This includes RV, ERV, TV, and IRV, or vital capacity plus residual volume. Alveoli are the primary sites of exchange of gases. Exchange of gases also occur between blood and tissues. O2 and CO2 are exchanged in these sites by simple diffusion, mainly based on pressure concentration gradient. Solubility of the gases, as well as the thickness of the membranes involved in diffusion, are also some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure and is represented as PO2 for oxygen and PCO2 for carbon dioxide. Partial pressures of these two gases in the atmospheric air and the two sites of diffusion are given. The data given in the table clearly indicates a concentration gradient for oxygen from alveoli to blood and blood to tissues. Similarly, a gradient is present for CO2 in the opposite direction, that is from tissues to blood and blood to alveoli. As the solubility of CO2 is 20-25 times higher than that of O2, the amount of CO2 that can diffuse through the diffusion membrane per unit difference in partial pressure is much higher compared to that of O2. The diffusion membrane is made up of three major layers, namely the thin squamous epithelium of alveoli, the endothelium of alveolar capillaries, and the basement substance, composed of a thin basement membrane supporting the squamous epithelium and the basement membrane surrounding the single-layer endothelial cells of capillaries in between them. However, its total thickness is much less than a millimeter. Therefore, all the factors in our body are favorable for diffusion of O2 from alveoli to tissues, and that of CO2 from tissues to alveoli.